morning. Today I am going to go over the Catalase Lab for you, just so I want to make sure you are fully understanding it, not just me talking to you through the screen. So I thought I'd make this short little video explaining the Catalase Lab. So it's ugly Christmas sweater day. Do you like it? Pretty funny. It was my mother-in-law's. So the substrate that we're working on is hydrogen peroxide, and the enzyme is catalase, which was in the liver or the potato, depending on whether you watched my video or you actually used a potato and peroxide at home. I was really excited to see a couple of them that worked really well. You got a lot of foam. So we started by placing liver in the beaker or potato in the beaker and then pouring our hydrogen peroxide over it. And hydrogen peroxide is H2O2, and we exposed it to catalase. This was either in the potato or the liver. And then we saw bubbles, and we saw bubbles because we're breaking the bonds in the H2O2, and the catalase is speeding that process up. And the bubbles was oxygen gas, bubbles, and eventually the liquid left behind would be water. So, right, the liquid left behind. If we left a beaker of hydrogen peroxide just sitting out, it would naturally degrade. It degrades easily. That's why it's in a brown bottle. So when it's in light, it will degrade easily. We just sped it up quick. We sped it up and it went really fast. And so you drew models, and I just kind of wanted to model a model. So we had H2O2 in the beaker, and this is our substrate, the substance that we want to break down. And then whether we did liver or potato, I'll say potato, either works. Maybe, you know, and I should add that over here. So it could have been liver or potato. Both have catalase. Liver just has a little bit more. So these are our potatoes, which this is our source of catalase, the enzyme. And when we did that, we saw lots and lots of bubbles, and we had foam build up. So those are oxygen gas, right? This is evidence, right, of change. We know the enzyme substrate complex is forming. We know they're joining at the active site of the enzyme. These two substances, catalase and hydrogen peroxide, we have evidence that this has taken place. The oxygen gas is evidence of change. With the liver, we also, this was hot, right? So you know that something is being broken down because energy is being released in the form of heat. So because we're taking apart this, a reminder, this is a catabolic reaction. Hope this was a helpful little video.